So you may have heard of interferon beta being used as a possible treatment for COVID-19. Now, what I want to do is go over what exactly interferons are and how viruses, or how explicitly coronaviruses, have learned a mechanism by which it tricks the body into not producing sufficient levels of interferon. And this is what allows the viruses to suppress the immune system locally so that they can multiply more and more. And it's not just suppressing the immune system of the white blood cells, but also the innate immune system of the cells nearby. Okay, so with this, I just want to point out the top stories regarding interferon. So it looks like there is promising treatment by giving interferon to people, and it sounds as if it's giving it to people early in the disease progression, so before they have really bad clinical symptoms. And part of this is because interferons, they're activating the immune system. They are convincing cells to they're convincing the white blood cells to damage more tissue, right? So this might not necessarily be a good treatment for after you've gone into a hyperinflammatory state where your immune system is already hyperactive. This might be something that will be more beneficial early on when your immune system is actually being suppressed. And during the suppression, the virus has time to multiply. So this might be something that should be administered early. If the clinical trials do show promise, it's more likely going to show promise in the early stages. And this is partly due to some of the deleterious side effects of using interferon treatment, where it can actually cause cells to die because it's convincing the cells that basically there's a really bad virus infection or a really bad viral infection nearby, and this can cause some of the cells to kill themselves if they are exposed to way too many interferons. Okay, but with that, let's move on to a little bit of the biochemistry and the cellular signaling that is going on. So interferons are actually a class of many different types of proteins. And I say many, but quite a few different types of proteins in different classes. And so they're a group of signaling proteins that are made and released by host cells in response to the presence of several viruses. So once a virus infects a cell, basically the virus will have components that make it up. Or I should say, it'll have components, or while it's multiplying, there will be certain windows of opportunity where they create things that are not naturally found within a normal human cell. And so your normal human cells have things that are scanning the cytoplasm, or they're scanning the insides of the cell, waiting to find these virus-like patterns. And once they find these virus-like patterns, then it activates signals that convince the cell, oh, there's a virus inside of me. I should warn the cells around me. And okay, and just so you know, these things that your cells look out for that alert it to a viral infection, these are called pathogen-associated molecular patterns, or PAMPs. So these PAMPs are what the cells look out for, but we'll go over more of that later. So these interferons are made by host cells in the presence of several viruses. In a typical scenario, a virus-infected cell will release interferons, causing the nearby cells to heighten their antiviral defenses. So it's basically telling the cells nearby, hey, I'm infected with a virus. Maybe you should start preparing yourself for protecting yourself for viral infection. So this is what would the word be? It'd be warning the nearby cells to be proactive in their response to a virus, rather than being reactive and only being able to do something after the virus has already infected. So interferons belong to a large class of proteins known as the cytokines. So molecules which are used for communication between cells to trigger the protective defenses of the immune system that help eradicate pathogens. So these cytokines are basically hormones, but they're hormones that are specific for warning the immune system to do one thing or another. So interferons are named for their ability to interfere with viral replication by promoting cells from virus infection. And interferons also have various other functions. They activate immune cells, such as the natural killer cells. And so these NK cells are a type of cytotoxic lymphocyte, which are critical to the immune system. So they basically kill cells that show any sign of virus infection, and I believe same thing goes for cancers. So the role of the NK cells is analogs to that of the cytotoxic T cells in the vertebrate adaptive immune response. So don't worry too much about that. So they will activate the natural killer cells, and they also activate the macrophages, which the macrophages, those are the white blood cells that very frequently will consume. They will undergo phagy, so they're large cells that can phage or eat other components in the matrix or the system. Anyways, they increase host defenses by upregulating antigen presentation by virtue of increasing the expression of the major histocompatibility complex antigens. So these major histocompatibility complexes, these are proteins that your cells produce. And what will happen is your cells will take their proteins, they'll take the proteins that are within the cell, and these proteins can become part of the cell through autophage or through endocytosis or through eating other things on the outside environment. It could also come from just being within the cell normally. And what happens is these proteins get degraded, and then peptides from those proteins get attached on to the major histocompatibility complex. And then these peptides are then released to the surface of the cell. Okay, and so basically the surface of the cell is presenting peptides that were found within the cell. And so this can help the immune system recognize when there's things such as cancer or things such as viruses. Because what ends up happening is in cancer, a lot of the proteins have mutated. And so if there's a really mutated protein, then the part that is mutated, if it is brought up to the surface, then the immune system won't recognize that mutated protein because, well, it's just a new protein, right? It's a protein that it's never seen before, and that way it activates the immune system to, hey, this is not good. I should probably attack and kill this cell because it has this new protein. So the same thing goes for viruses. If you get some virus protein particles in the cell somehow, some of those can be chomped up and then added to the major histocompatibility complexes, where when they are brought to the surface, it ends up activating the immune system, and by activating the immune system, it kills the cell, hopefully before the cell has produced that many viruses. Anyways, so these interferons can increase the expression of these major histocompatibility complexes, which would in turn make those cells more vulnerable to the immune system attacking them. And this could actually be bad, right? So theoretically, if it's dysregulated and you have too much of this being expressed, perhaps you could end up with an autoimmune response or cell death. The cells killing themselves. You know, there's a variety of things that could happen if this system gets dysregulated. Anyways, they also increase host defenses by upregulating these, and then certain symptoms of infection, such as the fever, the muscle pain, the flu-like symptoms, are also caused by the production of interferons and other cytokines. So these cytokines don't just make you better, they can often also cause you to feel sick. So you feeling sick is sometimes the result, or oftentimes the result, of these defense mechanisms that are being put in place. So these new hormones that are being produced in response to the pathogens, those will make you feel ill. Okay, so this, stepping back a little bit, this might be why so a lot of people who contract COVID-19 experience very little symptoms. So they could experience very little symptoms because SARS-CoV-2 is very effective at suppressing your immune system. And by suppressing your immune system, you end up not even realizing that you were sick in the first place. Let's continue. So, more than 20 distinct interferon genes and proteins have been identified in animals, including humans. They are typically divided among three classes: the type 1 interferons, type 2 interferons, and type 3 interferons. The interferons belonging to all three classes are important for fighting viral infections and for the regulation of the immune system. So, let's go to the types of interferons, and let's look into the induction of interferons as well. So the types. So interferon type 1, so all type 1 interferons bind to specific cell surface receptor complexes known as the IFN alpha beta receptors. So the surfaces or the cell membranes of all cells, or I believe all cells, probably just a lot of cells, they have these receptors on them. And these receptors are just waiting for the interferons to come in and stimulate them. And so these are the interferon alpha beta receptors, and they can bind to any type of the type 1 interferons. So And these interferon alpha beta receptors consist of the IFNAR1 and the IFNAR2 chains. So don't worry too much about that. So the type 1 interferons present in humans are interferon alpha, interferon beta, and this one is important because this is the one that is associated with the clinical drug, or the drug that is being proposed to help cure COVID-19 through a nebulizer. There's also interferon epsilon, interferon kappa, and interferon omega. So in general, type 1 interferons are produced when the body recognizes a virus that has invaded it. They are produced by the fibroblasts, which these are the extracellular matrix producing cells. So they are producing basically the structural integrity of the nearby area. That synthesizes, they are the biological cell that synthesizes the extracellular matrix and collagen. So yeah, they're producing collagen. So if you ever hear about collagen and stimulating collagen synthesis, those are the cells that are associated with that. And so anyways, they're produced by the fibroblasts and the monocytes, which are a subset of the white blood cells. However, production of type 1 interferon alpha is inhibited by another cytokine known as interleukin 10. So I believe interleukin 10 is one of the anti-inflammatory cytokines. Just know that that interleukin can inhibit interferon alpha. Anyways, once released, type 1 interferons bind to specific receptors on target cells, which lead to expression of proteins that will prevent the virus from producing and replicating its RNA and DNA. So overall, interferon alpha can be used to treat hepatitis B and C, while interferon beta can be used to treat multiple sclerosis. Okay, and so basically they're telling the cells, hey, there's a virus nearby, or hey, something's not right nearby, so therefore you need to hold it. You need to control yourself. Don't be producing a lot of proteins right now, because right now is not the time. Right now, a virus could actually have hijacked you, and you don't want to produce proteins. And also, prevent the virus from replicating its DNA. So downregulate polymerase activity if you can. So let's see. So then we have the interferon type 2s, and this is also known as the immune interferon and is activated by interleukin 12, which is one of those pro-inflammatory cytokines, if I'm remembering correctly. So these type 2 interferons are also released by cytotoxic T cells and the type 1 T helper cells. So Th1 is how I remember those. So however, they block the proliferation of type 2 T helper cells, and the previous results in an inhibition of T helper 2 immune response and further induction of the T helper 1 immune responses. So just know that it's there to help regulate what type of helper T cell response you end up having. So just so you know, it binds to IFNGR, which consists of IFNGR1 and IFNGR2. Okay. So and then there's interferon type 3, and I don't think we should really worry too much about interferon type 3. Just know that in general, types 1 and 2 interferons are responsible for regulating and activating the immune response. Expression of type 1 and 3 interferons can be induced in virtually all cell types. So pretty much all cell types have the ability to produce these interferons if they recognize that a virus is inside them. So this is generally good, right? However, here they were saying that interferon, what is it? In general, the type 1 interferons are produced when the body recognizes the virus that has invaded it, and they are produced by fibroblasts and monocytes, so that the fibroblasts and monocytes might be able to produce it more easily, but however, every cell seems to have the ability to recognize it. So how are these interferons induced, or how are they produced? So production of the interferon occurs mainly in response to microbes such as viruses and bacteria and their products. So binding of molecules uniquely found in microbes, such as the viral glycoproteins, viral RNA, and so this is the one that actually the SARS-CoV-2 and other coronaviruses have a mechanism by which to basically confuse the cell from being able to recognize viral RNA. And we'll talk more about that later. So the bacterial endotoxins, which are the lipopolysaccharides, and bacteria flagella, the CpG motifs by pattern recognition receptors. So these are the PRRs. So these PRRs, these are receptors or proteins that are able to recognize one of the things on these lists. And there are many different pattern recognition receptors. And so, and they are, some of them could be membrane-bound toll-like receptors, don't worry too much about that, and cytoplasmic receptors such as RIG1 or MDA5. So this MDA5, this is one that is going to be very important for us because this is a protein that can recognize double-stranded RNA. So for most or for you, almost all of the DNA, or sorry, almost all of the RNA within your body is single-stranded, in that there is only one strand, and it is predominantly used to synthesize proteins or be used as a template to synthesize proteins through a process known as translation. So the virus is hijacking this, or SARS-CoV-2 and other coronaviruses are hijacking the system because they are RNA viruses. They are also single-stranded RNA viruses. But there is a process by which they have to multiply. And in a small window of time during this multiplication process, you end up with double-stranded RNA. And this double-stranded RNA is not found in organisms normally, right? So since it's not normally found, it is a pattern that cells can recognize as a viral threat. So, and these things would be, let's see. Yeah, just know that these can all trigger interferons. So the toll-like receptor is important for inducing interferons in response to presence of double-stranded RNA viruses. The ligand for this receptor is double-stranded RNA, and after binding double-stranded RNA, this receptor activates the transcription factor IRF3 and nuclear factor kappa B, which I've also done a video on, or a series of videos on. I did actually 4. So, which are important for initiating synthesis of many inflammatory proteins. So these, once you see double-stranded RNA in the cell, activate a pro-inflammatory response, and this is a response that seems to be missing in a lot of coronavirus patients early on in their disease progression. So RNA interference technology tools such as small interfering RNA, you know what, don't worry too much about any of this. So that is how interferons are induced. So I'm going to go more into detail with this, let's look up interferon beta. So here's interferon beta, and then explicitly I'll go into this interferon beta 1 because, or IFNB1, because this is the current what would the word be, pharmaceutical that is going through FDA trials right now to be nebulized and breathed in to help activate your immune response. And this has helped to prevent you from having more serious complications down the road. So just know that it has antiviral, antibacterial, and anti-cancer properties. And the one that we're probably going to be looking at is either interferon beta 1A or interferon beta 1B. So interferon, these are basically the same. However, one is coming from, or is being synthesized from mammalian tissue, and one of them is being synthesized from E. coli. So let us look just to double check. So I don't see anything here regarding COVID-19. So it was the beta 1A that is being researched. So don't worry too much. You can look this stuff up for yourself if you want. I'm not going to really dig deep into fact-checking Wikipedia that much. Just there's clinical trials regarding the use of interferon beta 1A for SARS-CoV-2 and also MERS-CoV apparently. So, and just know that this is produced by mammalian cells. So we basically took the gene to produce interferon beta 1 and then inserted it into a mammalian cell culture where the mammalian cells are now excreting this interferon beta 1A, and this secretion, we use that we basically bottle it up and then inject it or nebulize it into people now. So, and that is in contrast with the interferon beta 1B, which is produced in E. coli cells. So, and this is just a bacteria. So that is what we are working with. Now, how do cells, or I should say, how do coronaviruses evade this? So you could imagine if you're a virus, this is going to be one of the defenses that you have to just deal with, or you suppress it. So you can either just deal with a hurdle or you can suppress the hurdle. And the more successful viruses are the ones that are successful at suppressing the host defense mechanisms. So here, I want to go over this research article. I'm not going to go over the whole thing. I'm just going to go over the abstract and the concluding remarks. So here, coronavirus endoribonuclease targets viral polyuridine sequences to evade activating host sensors. So what is an endoribonuclease? An endo, meaning this is inside, ribonuclease, this is something that will break apart RNA. So a coronavirus endoribonuclease is going to search RNA and destroy it, or at least take water and then cause hydrolysis of the molecule and break it up into smaller pieces. And this explicitly targets the viral polyuridine sequences. So these viral polyuridine sequences, so uridine is one of the letters that make up RNA, right? So RNA, like DNA, has 4 letters that basically are the code. And you can have these polyuridine sequences that are the result of that are the result of creating this double-stranded RNA. So this endoribonuclease targets these polyuridine sequences that are from the virus and destroys it. And by destroying a part of itself, it is able to hide a part of its identity. And by hiding a part of its identity, it is able to survive a little bit longer in society. So let us begin. So let's just go to the abstract. So the coronaviruses, these are positive sense, which means the strand that is injected into the system or into the host cell, this strand can be directly read by the ribosomes to produce proteins. This is in contrast with a negative stranded RNA, which will first have to be basically either reverse transcriptase into DNA, or it needs to have the complementary strand be synthesized first. And the complementary strand will be the one that has the proteins necessary to duplicate. Anyways, just know that coronaviruses are positive stranded or positive sensed RNA viruses, and they can emerge from endemic reservoirs and infect zoonotically, causing significant morbidity and mortality. The coronaviruses encode an endoribonuclease designated endo-U, and this is because it's an endoribonucleotase that attacks these polyuracil sites, and that facilitates evasion of the host pattern recognition receptor MDA5. So remember what I said earlier, this MDA5 is a particular protein, and I think I might already have it up. Nope, I don't. One second. So this MDA5, this is a protein that is within your cells. It's called a RIG1-like receptor, don't worry too much, but it is a double-stranded RNA helicase enzyme. So it is an enzyme that actually acts upon double-stranded RNA regularly. But it also functions as a pattern recognition receptor, or a PRR, and they are capable of detecting viruses. It is generally believed that MDA5 recognizes double-stranded RNA over 2,000 nucleotides in length. However, it has been shown that while MDA5 can detect and bind to cytoplasmic double-stranded RNA, it is also activated by a high molecular weight RNA complex composed of single-stranded RNA and double-stranded RNA. Don't worry too much. So for many viruses, effective MDA5-mediated antiviral responses are dependent on functionally active LGP2, and I guess this is just another protein, so the probable ATP-dependent RNA helicase. So yeah, it's dependent on this, and the signaling cascades in MDA5 and is initiated through CARD, through the CARD domain. So some observations made in cancer cells show that MDA5 can also interact with cellular RNA and is able to induce auto-inflammatory responses. So let us not worry too much about this. Just know that this is a protein that is recognizing the double-stranded RNA that is coming from the virus. So, and it seems as though this MDA5 specifically is targeting poly-U sites. Anyways, but the target of the endo-U activity was not known. So here we report that endo-U cleaves the 5' polyuridines from the negative sense viral RNA termed pun RNA. So the polyuridine from negative sense viral RNA. This is pun RNA. Okay, so which is the product of the poly A templated RNA synthesis. So when your mRNA, wait, okay, let's continue. So using a virus containing an endo-U catalytic inactive mutation, we detected a higher abundance of pun RNA. So in the cytoplasm compared to the wild type infected cell. So they took a cell and they infected it with viruses. One of the viruses has this endo-U activity mutated or destroyed. The other has it functioning correctly. And so they infect the cells and then they look at the RNA that is inside it and they said, oh, if we get rid of this endo-U activity, we end up having a lot, we end up having less, or I should say, using a virus containing the endo-U catalytic inactive mutation, they detected higher abundance of the pun RNA. So this pun RNA, which is a target by your pattern recognition receptors, there's more of these. So in the cytoplasm compared to the wild type infected cells. So furthermore, we found that transfecting pun RNA into cells stimulates a robust MDA5 dependent interferon response. So they discovered here as well that if you just inject the pun RNA, you are getting a robust interferon response. Okay? And that the removal of the polyuridine extension on RNA dampens the response. So if you remove the polyuridine from the pun RNA, you still get a response, but it is significantly less. So this is telling us that the polyuridine extension, this is one of the most important parts. So overall, the results of the study reveal that pun RNA to be a coronavirus MDA5 dependent pathogen associated molecular pattern. So this is the pattern on coronaviruses that this MDA5 recognizes. So we also established a mechanism for endo-U activity to cleave and limit the accumulation of this PAMP, or this pathogen-associated molecular pattern. And since endo-U activity is highly conserved in all coronaviruses, inhibiting this activity may serve as an approach for therapeutic interventions against existing and emerging coronavirus infections. And so what I also want to point out is this was first published March 20th, but this was received for review December 9th. Okay, so this was sent off for review before any of this pandemic even happened, and it was just accepted in March. So with that, let us go down here. So yeah, what I'm going to do is I'm going to skip all of this and just go down to the conclusion. So 1 second. So the discussion. And I also got to mention is for mRNA to be properly read by the ribosomes to produce proteins, you usually have a polyadenosine or a poly-A tail that is appended onto the mRNA. So if you look at mRNA, I'll just look at messenger RNA, and let's go to the Wikipedia. So usually at the 3' end, there is going to be a poly-A tail. So this is what's going on here. You have this mRNA that came from the DNA, but you have this polyadenosine tail that is added on to the end of it, and that helps with the translation process. Now, the viral RNA that is injected into you from the coronaviruses, they also get a poly-A tail, or they're already primed. I don't know if it's added onto it or if it's already there. You'd have to look it up yourself. But there is a poly-A or an adenosine-rich tail on the end of it. So what ends up happening is when the virus gets, what would the word be? What would the word be? When a polymerase comes and duplicates or creates the complementary strand for this mRNA, or I should say of the virus RNA, the virus RNA has a poly-A tail, and then this is going to become a poly-U tail for the complementary strand, because a lot of A's A's will bind to U's, right? So these poly U's are unique. Those are unique to viral proteins, right? And so that at least will be the recognition motif that this MDA5 is going to search for. So with that, just know that that is what's happening. So our study reveals that the coronavirus endoribonuclease activity degrades the PUN RNA. So the virus itself is going to destroy the poly-U nucleotide RNA. So, and which acts as the viral PAMP, or the pathogen-associated molecular pattern. So endo-U cleaves the poly-U sequence on the PUN RNA, limiting the length and abundance of the poly-U extension. This reduces the interferon and stimulating effects of PUN RNA, so which without endo-U digestion activates the host sensor MDA5. The fact that endo-U is highly conserved in all coronaviruses suggests that endo-U activity is important for sustained replication in the host. So if you can get rid of this, then, or I should say, if you can get rid of the endo-U nuclease activity, you end up with more of the PUN RNA. And the more PUN RNA you have, the more the cell is convinced it has a viral infection, and therefore will be producing these interferons. And this is, but the downside of this is that if it does have endo-U activity, the virus can exist longer in the cells without activating the immune response. And if they can last longer without activating the immune response, then they can duplicate more and more until that happens. So our study reveals the PUN RNA is a PAMP, and the endo-U activity is essential for limiting the accumulation of PUN RNA. So here, let's just show this example. So here we have the positive strand of the virus. And then the RNA-dependent RNA polymerase, so the RDRP, this is the virus protein that duplicates or creates the complementary strand of the original virus RNA. This will create these negative sense RNAs that are supposed to be complementary with this. Regardless, these are negative sense, and they normally will be read again by the RNA-dependent RNA polymerase to produce more of this original sequence. Okay. Now, the problem with this is if you have endonuclease activity, this endonuclease is going to remove these poly-U extensions, right? So they're going to remove this. And so you end up with these short U sequences that do not function as PAMPs and therefore do not get recognized by MDA5. And then by not being recognized by MDA5, you get no interferon signaling. However, if you have an endonuclease U mutation, then in that particular virus, these PUN RNAs are still around, right? And so these poly-U areas are actually going to basically curl in up on themselves, apparently, especially if there is an adenosine or a guanosine rich domain. And by doing that, it ends up creating double-stranded RNA that recognizes, or at least has the cell recognize that this is virus, and therefore I need to destroy it. So that's remarkable that this is the method by which it happens. So, and I think, yeah, there's not really much reason to go over anything else. So just know that this is why interferon is being considered, interferon 1 is being considered as a treatment strategy for the early stages of the virus life cycle, because you want to stimulate the immune system early. That way the virus doesn't become out of hand. Now, granted, what might be problematic is in the future, and you know what, let's see if I interferon beta 1a. So you can have side effects such as if you inject this, it can form skin reactions such as, or at the injection site, that may include cutaneous necrosis, or the dying or the killing of the stem cells, or killing of the skin cells. So skin reactions with interferon beta are more common with subcutaneous administration and vary greatly in their clinical presentation. So one of the side effects is if you inject interferon beta, you end up basically convincing those cells that they are infected with a virus, or they're close to it, they're infected with the virus, I need to shut down everything. And if this becomes dysregulated, you can cause things such as necrosis or cell death. And that could also be associated with the immune system becoming hyperactive or overactive in that particular area. So this is something that must, I would presume, maybe I'm wrong, but I would presume that it would only be beneficial early while the virus is still multiplying. It will not be good when your immune system is already overactive and causing severe illness. So the point is avoid the severe illness rather than try to do damage control later. So with that, I'm going to end this video. I think this is good enough for us to have a general understanding of what is going on. So with that, thanks for watching. If you have any questions, leave them down in the comments, and I will see you tomorrow.